Hello everyone. Due to some technical difficulties today, I don't know why I can't get my program to work. We're just going to do the video this way. I'm sorry you can't see my face this time. But we're just going to do it this way, and then hopefully by tomorrow I can get this figured out. So last time we were covering the nerve cell and we were looking at the axon. So we had seen the dendrites, then we had seen the cell body with the nucleus and all the organelles and everything that it does to make sure that the cell stays alive. And we went down to the axon. We said the axon is where that message, that nerve impulse takes place. It's where that transmission between the dendrites and the synaptic knobs occurs. So when it comes to the axon, what you're going to notice, we said, that is that it has these little myelin sheaths, these little sausage links that cover it up right here. And so we're going to talk about myelin next. And we're going to do it as a sort of a subpoint here. So a part of the axon is that myelin sheath, which is a lipid covering that is found only on some axons. So not all neurons will have this myelin sheath covering. These kind of axons are said to be myelinated. So you would say myelinated neurons or myelinated axons. Okay. And what does this myelin sheath do? First of all, it acts as a protective layer. And also, it speeds up the sending of the message, or we could say the neural impulse. Okay, so it speeds up that message, and we're going to see in just a second why that is. Okay, and also I'm gonna attach a video where it talks about multiple sclerosis, which is a disorder that destroys the myelin sheath and how that affects the nervous system and how it works. Okay, so but before we get to that, each of those little sausage links is called a Schwann cell. So each link is known as a Schwann cell. Okay, and that's the name of the scientist that discovered it. So what we can do is we can say each one of those is a Schwann cell. Okay, the entire covering, this whole thing right here, is the myelin. Right, so we can say myelin sheath or myelin. So the entire thing is the myelin, and then each one of these guys right here is a Schwann cell. All right, and again, it's made of lipids. Now the space between Schwann cells are known as the nodes of Ranvier. And I should probably capitalize that because it's the person again after the scientist. So those are known as the nodes of Ranvier. So again, if I go back up here, oh shoot, I probably should not have picked purple. I'm going to change the color there. Color, and we're gonna go that. And so here, oops, this space between the little sausage links right here, those are the nodes of Ranvier. It's the part of the axon that is exposed. Okay, so don't forget that whatever is running through those little sausage links through the Schwann cells is the axon. But the parts of the axons that are exposed, that are not covered in myelin, those are known as the nodes of Ranvier. And the reason why these are going to be important is, again, in order for it to speed up the transmission of the impulse. And the way I like to explain this is if I had my axon and the axon is not myelinated, then what's going to happen is the message, let's say that the message we had it, which color the message? I think it was red. Yeah, so the direction of the impulse, it was in red. So the message is going to have to travel every single section of this neuron. And so if you were to walk across the room heel to toe, it would take you quite a long time to send the message. So this is the message traveling through an unmyelinated, unmyelinated neuron. Now look at what happens. So we're going to say, look at what happens when you have a neuron that is myelinated. So now we said we're going to have those Schwann cells. So here's one, there's the other, okay? And by having this fat covering, it's almost like a wire when a wire is insulated, right? And it's like, don't forget that this is an electrical impulse. And so it's almost like the electrical impulse is insulated from those little sections. And so then what happens here is my signal, my message, is not going to go heel to toe. Well, it'll go like this, but then when it gets to the node of Ranvier, it'll jump. And so this makes the transmission a lot quicker, okay? So this is message in myelinated neuron. So it's much quicker because it jumps the nodes. It does not go, that message doesn't have to travel through those particular nodes. And so this type of transmission right here, this is known as saltatory conduction. Okay, saltatory meaning to jump. So that's saltatory conduction. So now if we look at a disorder like, for example, multiple sclerosis, with multiple sclerosis what you have is you have your immune system that destroys the myelin. It gets rid of these particular coverings. And so what you have is you have a neuron that was previously transmit information very, very quickly. Every time that I have to send a message, it became quickly that it has to do this sort of transmission. And so your neural processes are slowed down and impaired quite a bit. So that is the symptoms of multiple sclerosis. And depending on which neurons are being attacked in the body, that's why multiple sclerosis symptoms can vary so drastically between people. Okay, is it a speech impediment? Is it a movement impediment? It depends on what parts, what neurons in the body are being attacked by the immune system. Being an autoimmune disease, it's being studied quite a bit, just like allergies and just like Crohn's disease and colitis. Rheumatoid arthritis is another kind of autoimmune disease. We don't know why the immune system attacks your body like that, but it's being studied quite a bit. Okay. Now, these kinds of neurons right here, the ones that are unmyelinated, these ones can be found in what is known as gray matter. So I don't know if you've heard that before, gray matter versus white matter when it comes to your nervous system. And the reason for that is instead, the ones that are myelinated, because of that fat covering, they look white, almost like it's more of a yellow, but they're known as white matter. So if you ever look at a picture of a brain, whoops, we're going to go brain, white versus gray matter. You're going to see the difference, okay? When I look at a brain, you're going to see the difference between gray matter and white matter. The very outside of the brain has that gray matter and the very inside has the white. Over the next few weeks, once we get to the brain, I'm going to do a virtual dissection for you guys so you can see the different parts of the brain, and you should be able to easily see the white versus the gray. And I'm not going to show it to you right now because I want you to see that one. That's why I'm showing you only like these cartoon pictures, but that's what it would look like. Okay, so gray matter means the neurons that are part of that section don't have any myelin covering them, whereas white matter, the neurons in that section have myelin, and therefore the transmission is a lot quicker. So in this section right here, the gray matter, the transmission is a little slower. Thank goodness for a lot of these places there isn't a lot of distance to travel, so that won't make much of a problem for it. Okay, like I said, there is a video on multiple sclerosis if you want to watch it, but that's what I'm just going to make a note here. MS, multiple sclerosis. There's a video that you can watch, but it is autoimmune disorder that destroys myelin. And so you can sort of see what the problem there is. Okay, so we've made our way from the dendrites to the cell body down the axon, and now we're getting to these little things called synaptic knobs. So that's the last section that we're going to talk about for our neuron. Oops, wrong purple. Synaptic knobs. Okay. Now the synaptic knobs, they contain chemicals. We said that this process is an electrochemical impulse. So we are going to look at the electrical nature in just a second. We sort of gave an idea up here, but the synaptic knobs is where the chemical piece takes place. And these chemicals are known as neurotransmitters. And they help not with the communication within the neuron itself, sending the message within, but with the communication between different neurons. So they help with the impulse, the message traveling to the next neuron. Because here's the thing that is actually really cool. Neurons don't physically touch. So as much as they are in communication with one another, they don't physically touch. And the reason for that is we're dealing with electricity. And so you don't want an electric shock to happen by accident. And so the way to prevent that is by making certain that these neurons are not actually touching each other. So when I have one neuron that we've drew above, okay, and I can put myelin around it or not, and then I can have another neuron with their little synaptic knobs. What we see is that the dendrites of one and the synaptic knobs of the other don't physically touch. What they use is these little chemicals called neurotransmitters to cross that gap. So, in other words, when my message comes in from the dendrites and makes its way to the synaptic knobs, in order for it to go to the next one, I need those neurotransmitters to make the transition between one and the next. And then once the neurotransmitters bind with the next neuron, the next message is initiated. The next one is started. And we'll talk about that in a little bit as well. Okay? Now, when it comes to neurons, the next thing that we have to look at is different types of neurons. So we're going to base this on structure and also based on function. And in a way, just by drawing them, it's going to be based on looks as well. Okay, and we're going to start with a sensory neuron. That's structure. And then interneuron. And finally, we're going to have motor neuron. So a sensory neuron, what you're going to see, actually, you know what? I'm actually going to start with a motor neuron because guess what? You've seen one already. So the one that we labeled together is a motor neuron. And you can see dendrites, axons, synaptic knobs. When it comes to sensory neuron, again, same structures. Like I said, those are going to be the same for everybody. So I'm going to have dendrites, but then I'm going to have a little piece of axon. Then I'm going to have my cell body looking a little weird over there. Almost like a periscope in a submarine. And then I'm going to have my synaptic knobs on the other side. And when it comes to an interneuron, again, I'm going to have my dendrites and axon, usually pretty short for interneurons, and then usually unmyelinated for interneurons, and there's my synaptic knobs over there. Again, the direction of impulse is always from dendrites to synaptic knobs, dendrites to synaptic knobs. That doesn't change. So they do look different, okay? And also based on function, they're going to be different. When it comes to sensory neurons, as the word said, sensory, they pick up information from the senses. So they pick up info, or stimuli is the correct terminology, from the outside world or from the body, from the inside too. And they send it to the central nervous system. They send it to the brain for interpretation, right? What do I do with this information? That's what the sensory neuron does. It picks up all that information and sends it to the central nervous system, okay? Interneurons, these guys, they transmit information just within the central nervous system. So for example, these guys would be the communication link between the brain and the spinal cord. Or you could just say communication within the brain. There's different parts of the brain that communicate with one another. And so the way that the brain communicates with other parts of the brain, with itself, is through interneurons. And then a motor neuron. So these guys are going to carry responses from the CNS. Oh, pencil didn't like that. Okay, from the CNS to what are known as the effectors. And effectors could be cells, glands, muscles, organs, whatever that message is, it needs to be sent to a part of the body to do. Okay, so we talked about, I said, you know, I could have an itch on my neck, And so that's sensory information being sent to the brain. And then the brain says, okay, there's the itch on your neck. It's communicating through interneurons and saying, was there a time where we already had an itch on the neck? What did we do with it? Or was there an itch at a different part of the body? And how do we respond to that? Could we apply that same mechanism to this situation? And then the brain sends the information to my motor neurons and saying, okay, try ahead and scratch it, go and see what happens. And by scratching it, then that information is sent to the brain, whether the issue's been resolved or not. Okay, so that's the difference between your neurons. Okay, now that we've seen the different kinds of neurons that are present in the body, I want to talk about how they communicate. So that I want to sort of see the nerve impulse. We're not going to go into a ton of detail when it comes to the nerve impulse, but I do want to show you some information about it. I want to show you sort of what happens throughout the body. And with this, what we have to do is we have to look at our axon, because that's where the nerve impulse takes place. So the function of the neuron depends on the nervous impulse, depends on both electrical and chemical events. Now, we're going to talk about electrical events first. And don't forget that electricity is just a flow of ions. In the case of the electricity that lights up your room or turns your computer on, what you're dealing with is you have a circuit, we have wires, and we have a flow of electrons. So again, those ions are negative ions that are being passed along that wire. The circulation of those ions back and forth is going to cause the electricity, the energy to then light up the room or turn on the computer. When it comes to a neuron, we're not dealing with electrons, we're not dealing with negative ions, we're going to deal with positive ions. But again, I'm not saying that electricity is a flow of negative ions, I'm saying any ions. As long as we have a flow of them, we're going to have some sort of electricity. Okay, so here, what we do have is we have an axon membrane. Actually, I'm just going to erase that. There we go. So we have an axon membrane, and we're going to label it. This is the outside of the axon. This is the inside of the axon. And this sort of area of where neurons are going to be, so sorry, this is the axon. I'm just going to write here axon. It's flooded with different chemicals. And you're going to have, for example, sodium ions, and I'm going to use little circles for sodium ions, and I'm also going to have potassium ions, and I'm going to use triangles for that. And you're going to see, you're going to see that there is all sorts of chemicals everywhere, but these two are the ones that are important. Sodium and potassium are important, and we know sodium and potassium are both positive. Okay. Now, your body spends a ton of energy. Almost a third of the energy that you get from your food is spent to power something called a sodium-potassium pump. And a sodium-potassium pump has a job. And as you can see here, it's got a specific shape to it so that it fits particular chemicals in it. Oh, look at that, sodium and potassium. So that's why it might be called sodium potassium pump. So this is a sodium potassium pump. And the job of the sodium potassium pump is all day long, what it does is it pumps 3 sodiums out of the axon and 2 potassiums inside, constantly. And again, I'm going to attach a video here, so I'm going to put video for you guys to watch as well. It's attached, it's going to be attached to your notes. So there is a sodium-potassium pump. I might even attach to this one if we have time. Sodium-potassium pump, constantly pumping 3 sodiums out of the axon and 2 potassium inside. What this does is it creates something known as a concentration gradient. A concentration gradient is needed for transport in a cell. So I don't know if you remember from Science 10, concentration gradient, if I have more water molecules outside of a cell and less water molecules on the inside, through osmosis, that water will move into a cell. So what I've done is I've created a concentration gradient where I have more sodiums outside of the axon and less on the inside. So what the axon is going to want to do is going to want to transport those sodiums in and those potassiums out. It's going to want to even things out. And as I have a flow of ions through the membrane, that's going to create electricity. And that electricity is going to travel down the axon. So I'm going to show you, there is a little video, a little clip here, a little cartoon. Okay, a little diagram. And so you can see that this is the axon. And so if I separate my axon into sections, so there's a section, there's another section, there's another section, you can see here that what's going to happen is you're going to have this particular section here. This is when the sodium ions are being brought in. And then here, potassium ions are being brought out. And then again, sodium ions are going to be brought in, potassium ions are going to be brought out. So I'm going to have this flow of ions continuously through my through the axon. Okay, so let's see if I can get that little video that I already, I think, loaded up onto your Google Classroom. It's just a quick clip, and it's actually really good. This one, it's only like a minute. Oh, will it let me? It won't let me. Okay, I'm just gonna go nerve impulse transmission. I want to go on YouTube. There it is. Out, in. You can see sodium ions and then they return out. Sodium ions pulled out, then pulled in. And it is that constant pulling in of those sodium and potassium ions that is going to cause electricity. So it's that constant flow of ions in and out. Okay, so here what I have is sodium ions being pulled in. Okay, and then here in the repolarization stage, this is where the potassium ions are being pumped out, because what I want is I want them to both be even. And if I always have more of one on one side and more of one of the other on the other side, the cell always wants to get even. And when it gets even, that's called resting, when it's trying to get things back to normal. And then now we'll move on to the next section. So now sodium ions are going to be pulled in, and then potassium ions are going to be pushed out. And then I get to that next section of the neuron, so I'm over here now. And then again, sodium ions being pulled in, and the process repeats itself. And that's how we get that message being sent down the axon. Okay, so that's that. We're not going to go into a ton of detail, like I said, when it comes to the action potential, but you can see that when sodium ions are being pulled in, that's called depolarization. And when potassium ions are being sent out, that's called repolarization. And then, like I said, when the neuron is not actually firing, it's not sending a message, is known as rest. Okay? Now we get to the end of the neuron. So once we get to the end of the neuron, we are looking at the synapse, okay? So now in the synapse, I'm just gonna choose a different color here. I'm gonna draw you the synapse again. Actually, we don't even need to do that. All I need to do is take this and go copy. Nope, it might not, there we go. So this is the synapse, and this is the area right here that I want to zoom in. So what we're going to see here is we're going to see the synaptic knob of one neuron and then the dendrite of the next. All right, here we're going to have some mitochondria because I'm going to need some energy. I'm also going to have some vesicles because those vesicles are going to be filled with neurotransmitters. So these are vesicles with neurotransmitters. These are mitochondria. For energy, I'm going to need some ATP there. And then on the dendrite side, on the other side, what I'm going to have is I'm going to have these different, these are known as receptors. All right, so I have the synaptic knob of the presynaptic neuron, so that's the neuron that comes before the synapse. And then I'm going to have the dendrite of the postsynaptic neuron, that's the neuron that comes after sort of that little space. And by the way, the space here, this is known as the cleft or the synaptic cleft. That's the space, okay, in between the two neurons. So what's going to happen here is as my impulse, as my electrical impulse arrives here, it causes the vesicles to be pushed against the membrane of the synaptic knob. And when that happens, with the help of some energy, ATP, if you remember exocytosis, the neurotransmitters are released in the cleft, and they diffuse through the cleft until they arrive at the receptors. And if the neurotransmitter fits with the receptor, then what it does is it causes the next neuron to have that impulse, that electricity, that exchange of sodium and potassium ions to occur. How does that happen? So there's two things that can happen here in this kind of scenario, and it depends on the kind of neurotransmitter. And we're going to end it at this section. So when it comes to neurotransmitters, they can be excitatory or they can be inhibitory. So excitatory doesn't mean that they make you excited. It just means that they cause another message to be sent. So they cause another impulse to happen. So they cause an impulse to happen. And so in that case, the neurotransmitter binds to the receptor and it causes sodium ions to rush into the axon. And we saw from our little animation that whenever we have those sodium ions rushing into the neuron, we have that depolarization. We have that nervous impulse starting, okay? So that's an excitatory neurotransmitter. An inhibitory neurotransmitter, it causes the impulse to stop. So it stops it dead in its track. It doesn't allow it to pass on to the next neuron. So instead of saying causes the impulse to stop, you can say it inhibits the impulse from happening, right? So it prevents the impulse from happening. How does it do that? So the neurotransmitter, this particular neurotransmitter, binds to the receptor and causes potassium ions to rush out of the axon. And so what we're doing is, in that case, we're getting to this second stage, and the neuron is kind of confused. It's like, well, now I have too many potassiums out. What happens to my sodiums? And so the impulse doesn't occur. We say that the neuron is hyperpolarized. It has way too much of a certain charge on one side, and therefore it does not transmit the impulse. Again, I'm going to put some videos up for you guys if you want to look at more detail, but for the purpose of this, like I said, we're not covering everything to an extreme detail, and so that's what we're going to leave it at for here. So depending on which neuron leaves this area right here, you're either going to have the impulse that goes through, and so I'm going to get a signal being sent, I'm going to get a response, or if it's a different neurotransmitter that binds to the receptor, then I don't get any impulse being sent. And so that would be good for things like painkillers. So pain signals are coming through, ow, 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 I have a headache, and then all of a sudden, a neurotransmitter, or in the case of taking an Advil, it could be a drug that simulates a neurotransmitter, binds to the receptor of my pain neurons and stops pain from being transmitted. And that's how sort of these painkillers work. All right. Now in the next class on Friday, what I'm going to do is I'm going to talk about different neurotransmitters and how they work to affect neurons and our brain and the signals that they send. And then we'll continue from there.